As a respiratory illness, COVID-19 is hard on the lungs. For very sick patients, that often meant relying on a ventilator to get through the worst. But even in milder cases, the effects can linger, sometimes for months. Joining us now for more on the line from Kingston, Ontario, Dr. Stephen Archer, professor and head of the Department of Medicine at Queen's University. And Dr. Gerald Evans, attending physician in infectious diseases and internal medicine at Kingston Health Sciences Center and chair of infectious diseases and professor of medicine at Queen's University. Welcome to you both.
Thanks for having us, Jeyan.
All right, so Gerald, I'm going to start with you. Of course, you guys both have a newly released report, but I want to know exactly what we knew about COVID-19 and lungs prior to that. What did we know? Did we know enough?
Well, we certainly knew that the major serious manifestation of this disease was a pneumonia. And that's because the original virus, the ancestral virus that emerged first in Wuhan, and then ultimately Alpha and Delta variants, tended to cause a severe infection in older individuals, and it was principally a problem with the lungs. So it caused pneumonia, and that resulted in some significant challenges with maintaining your ability to oxygenate your body. And so, as you pointed out, people who had that illness oftentimes required a ventilator to manage them, at least through those parts of the illness.
All right, Stephen, I want to come to you. Of course, this research team encompasses a team across this country. When was the research team assembled? When did this project really get going?
Well, it's interesting, the origin story of this, the universities oddly closed down across Canada. And I think one of the things I'd like to get across is research is an essential service in Canada, and we need to be at the bench. So universities in about March, when the first announcements came out about the pandemic, closed down for in-person use. So we were anxious to get our team back to the bench, as they say. And so in the team, part of it already worked with me in my research lab at Queen's University. And then I talked to Gerald Evans, who's an expert in infectious diseases and virology, and got some advice about how we might turn our cardiac lab that studied hearts to study this disease. And then we brought in Gary Levy from the University of Toronto in April of 2020. And then Shay Colpitz, a virologist here, in June of 2020. And then finally, our colleagues out in Saskatchewan at VITO, which is one of the research centers set up to handle the SARS-CoV-2 virus. We brought them in 2021. So that's the timelines of our teams of 24 people being assembled.
And Stephen, so you mentioned some of the members there. Give us a better idea of the scope. How big was this team, and what are the sort of the institutions that were involved here?
So 24 people at least. 24 people are on the paper that published the discovery, including Dr. Evans. And it's based at Queen's University, but with really important contributions from VITO, which is the Vaccine and Infectious Disease Organization in Saskatoon at the University of Saskatchewan. My colleague there, Dr. Aaron J. Berengy, and then Gary Levy at the University of Toronto. So 3 universities, 24 people.
All right, so Gerald, I'm going to come to you. When we talk about the actual study and the actual coronaviruses we're using, what viruses did we use?
Well, initially, we actually used a coronavirus called mouse hepatitis virus. And many, many years ago, I worked with that in Bob Anderson's lab at University of Calgary when I was a research fellow. And that causes a pneumonia in mice. So it was a model that we could use to look at that. And what the real advantage was is that we didn't have to have a high containment lab to work with mouse hepatitis virus. But ultimately, then it really had to move on to closer models, and then we moved to a human coronavirus called OC43. And then ultimately, as my colleague Dr. Archer mentioned, we were able to do work with SARS-CoV-2 through the VITO organization, which has a high-level containment lab, which allows us to work with that particular pathogen. So depending on how nasty these are to humans, they have to have various levels of containment in laboratories that work with those viruses.
All right, Stephen, I'm going to stick with you. In understanding and better understanding sort of COVID-19 and lungs, I think, as you mentioned, something we need to understand is low blood oxygen. You know, a lot of images of people in hospitals or even at home who have asthma or COPD will use an oximeter to sort of measure those levels. And I'm curious, can you break it down? What is technically low oxygen? What is low blood oxygen?
So doctors call low blood oxygen hypoxemia, just a fancy word for low blood oxygen. And basically, the whole purpose of your lung is to take in oxygen from the environment, and that allows you to make energy in your body. So if you don't have enough oxygen, you can't make the energy molecule ATP, and organs begin to fail. And in coronavirus infections, all three of those viruses that Gerald mentioned, they all cause hypoxemia and pneumonia. And what was interesting about the COVID pneumonia from the very beginning is doctors were noticing oxygen levels were lower than they were used to seeing with equivalent pneumonias caused by other bugs. And they quickly found out that this was due in part to the fact, the lung, most people don't recognize this, the lung has its own circulation called the pulmonary circulation. And normally it has an oxygen sensor. So if you have pneumonia in the left lung, it diverts blood to the right lung. But something was happening in COVID pneumonia that blood was going to areas that were full of pneumonia and thus couldn't take up oxygen. And so patients were coming in and they had two features there. Oxygen level was very low, which might land them on a ventilator. And the other thing is at least some of these patients weren't as concerned and short of breath as you might expect with a typical pneumonia. So we began to recognize this was somewhat unusual. And we wanted to understand why didn't they sense the oxygen and what was killing the lung cells in about the 5% of people that wound up in trouble with COVID-19.
I'm hoping you can get us some idea of what percentage of people who get COVID suffer from low blood oxygen, Stephen.
And Gerald can weigh in on this too if you want, because he's been our go-to guy throughout this pandemic. But basically, there's about 44 million Canadians have been infected. That's probably a gross underestimate. And about 47,000 people have died. So about 1% of people die. About 5% wind up in hospital, acknowledging that we probably only detect 1 in 5 or so infections. Wouldn't you say, Gerald?
Yeah, those are pretty accurate numbers. And I guess it's also important to point out that during the sort of first year and a half of the pandemic, we were dealing with variants and the original virus, which tended to produce this more profound picture that was studied. Since Omicron's come out, we know that Omicron, that variant and all of its subvariants, tends to cause infection more in the upper respiratory tract than the lung, although we still see lung damage and pneumonia occurring, particularly in elderly individuals. And, that was really the crux of trying to understand it. And I think many people can remember early in the pandemic, there were these really kind of worrisome photographs and film that came out that showed people collapsing in the street with COVID.
Right.
And that was essentially one of the really important things that this research answered, which is why do people not perceive that their blood oxygen is low to the point that they actually collapsed without sort of having any symptoms? And this was really critical to understanding because it was a remarkable thing that we saw early on in the pandemic.
Gerald, I want to stick with you. Of course, as you mentioned, there are different demographics here we're dealing with. Some people have healthier lungs than others. Are people with underlying health conditions like asthma, like COPD, more likely to be hospitalized, or is everyone at risk here?
Well, I think the most important thing, and I can't emphasize this enough, age is the critical factor for the severity of COVID. The older you are, the more severe a form of illness you're going to get. Interestingly enough, there have been studies which have looked at asthma, looked at chronic obstructive pulmonary disease, or COPD as it's called, and they've been kind of somewhat contradictory, probably more so with asthma. Asthma patients, surprisingly, at least in large studies that were published back in 2020 and 2021, don't seem to do as badly. Patients with COPD, who oftentimes have more than just airways disease, but structural lung disease, they can do worse. And especially we know COPD is a disease which tends to occur in older individuals, which again is the primary determinant of more severe illness.
All right, Stephen, before we get deeper into the science, help us understand why it's so important to understand what's happening on a cellular level when it comes to COVID infections.
Well, I think it might be tempting to think that we have vaccines and we have drugs, so why do we need to know any more? We know it's a viral infection. There are several things that I'm sure you're aware of, though, that tell us that we need to perhaps find other tools to treat these infections. First, this is the third pandemic we've had with the coronavirus in just 20 years. So we had SARS, we had MERS, and now we have COVID-19. So this type of virus, which is actually a pretty widespread virus, is going to probably come back. So we need a good understanding of how it actually damages the heart and lungs if we want to combat it. The other thing, too, is we learned a lot about ourselves as human beings, and 20% of our neighbors will not take a vaccine, and worse in other countries. And we also learned that Canada doesn't make drugs, so we're sort of downstream from the largesse of other countries in terms of getting these things. So we need some better understanding of the virus so we can prevent its ravages in the people that are most susceptible. And we need to recognize that there's not going to be just one tool to prevent damage from the virus. And furthermore, the stuff that we discovered in this paper, interestingly, seems to apply to most coronaviruses, including prior viruses like the virus that caused SARS. So there seems to be some commonality in this mitochondrial mechanism that we have uncovered.
Well, I'm going to stick with you and let's talk a little bit about that. And I want to make sure that we get, what was the central goal of your research when we talk about not only the pneumonia aspect, but also on the cellular level?
So we wanted to discover two things with our research. One is, why was the lung so whited out on the chest x-ray? What caused that lung injury, which is called diffuse alveolar damage? And the second is, why was the oxygen level so disproportionately low in the lungs? Those were our two goals.
Gerald, I'm going to come to you. You touched on this a little bit, but what distinguishes COVID patients with pneumonia from non-COVID patients with pneumonia?
Well, principally, if you look at adults, most pneumonias are actually bacterial. They're caused by bacteria, not viruses. So it's not that viruses can't cause pneumonia in adults. We know that influenza does, for instance. And in some people, even viruses like RSV and rhinoviruses, which cause the common cold, can cause pneumonia. But what's unique about viral pneumonias in general is that they tend to affect all of the lung. Bacterial pneumonias oftentimes may be sequestered in just one part of the lung, what we call a lobe. So a lobar pneumonia is very characteristic of bacterial pneumonias in adults. And the other thing, of course, is that the degree of hypoxemia, the degree of oxygen loss, although it's seen commonly across most pneumonias, it's very, very severe with some viruses like influenza and like what we're seeing with the coronaviruses in general. So our understanding of how that happens and is really critical to our developing therapeutics and also understanding what ultimately is happening in the lung over the long term. Because as you commented on early in the preamble, is that, you know, the effects of COVID after they've infected someone may persist for a while. And some of that is going to be due to damage in the lung. And understanding that is absolutely critical. And that's what the real key to this study has shown. And I would say it really underscores why we have to understand how viruses cause disease, or what we call viral pathogenesis, because that's a fundamental piece of understanding we need to have in order to move forward with ability to develop therapeutics, for instance, and figure out, you know, how do we as physicians treat patients who have this infection.
Stephen, I'm hoping you can break this down for me. Someone who has COVID-19 or is recovering from COVID-19 is having issues breathing. What does that look like when they are taking, you know, some air and oxygen into their lungs, and how does that sort of work in terms of their lungs and where the oxygen is actually going and getting oxygenated?
Well, if you start with like what we know about the pathology of this disease, in other words, what the tissues look like, their lungs are partially full of liquid. So oxygen doesn't go through that liquid very well. They're also a little bit scarred or fibrotic, so their lungs are stiff. And so when you combine all, and they're very inflamed, lots of white cells of various types of immune cells have infiltrated their lungs. So every breath they take is much more work than it should be. And whether they sense that work or not, they eventually get tired. And what starts happening is their oxygen level goes down. They become what we call cyanotic, which means purple like a grape. And at some point, if they don't get assistance, they will stop making energy and they'll get heart disease, lung disease, and they'll wind up on a ventilator. I should mention this virus doesn't just infect the lungs, although that's its number one target. It's perfectly happy to bother your heart. It can affect your kidneys. Your intestines are a big site of coronavirus infection. So all these organs begin to fail. So the person is not only hypoxemic, but they're suffering from various other forms of organ failure that can threaten their life, and both acutely and then in the longer term. And I should mention also, and maybe we'll get to this in the basic science, Jeyan, but the viruses have a long history of not liking bacteria. They fight each other. There's a long-standing war between viruses and bacteria. And our mitochondria inside our cells once were bacteria. So we're sort of bystanders in this war between viruses and bacteria, with our mitochondria being, to some extent, mistaken as bacteria.
So we'll touch on the history, but I want to stick with you in terms of how the coronaviruses damage mitochondria. What does that actually look like?
So I like to think of it like alien versus predator. These two nasties are fighting with each other. The virus comes in and it got RNA as its code, and it basically takes over our cells and uses us as to make a sort of like a photocopier to make copies of itself. But an interesting thing it does, and I don't know whether the virus intended this or not, but its code basically turns on a lot of our genes or turns off a lot of our genes, which target our mitochondria and our lung cells. And it also makes proteins which are part of itself. It doesn't have very many proteins, but these fundamental building blocks, some of them go right to our mitochondria and other critical structures in our cells, and they engage in what's called protein-protein interactions, and they often block the normal function of the cell. So they use our nucleus, our genetic code against us, and they also make their own viral proteins, which are toxic to our cellular proteins.
And Gerald, I'm going to come to you in terms of adding onto that. Were these the findings that you sort of discovered? Have we seen this in other sort of, not only coronaviruses, but other viruses in terms of how the mitochondria sort of react to these viruses?
Yeah, no, it's very fascinating because when Stephen and I first started our discussions about this long ago in the early part of the pandemic, actually I found a few papers that discussed the role of mitochondriopathy or disease in the mitochondria and in other viruses, particularly coronaviruses, but other viruses as well. But sort of the really fundamental understanding of how that happens is really part of what this study was all about. And I think this study has really expanded that area to make us understand how important that kind of interaction is from other viruses and how it very severely affects cells. If cells can't make energy, as Stephen was mentioning, they're going to die. And one of the things that's really interesting, again, about the studies that Dr. Archer's done for many years with mitochondria is understanding how dysfunction and damage to the mitochondria eventually results in cell death, or what's called apoptosis. And that is fundamental to us understanding reversibility or irreversibility oftentimes of various virus infections.
All right, Stephen, I want to come to you. You talked about alien versus predator here, and I want to get through some of this history. There's an interesting, as you mentioned, evolutionary angle to this. Talk to us a little bit about how viruses, bacteria, and mitochondria are all sort of linked in this when we talk about COVID-19.
So I'll go back to 1967, which was not only Expo in Canada, but it was also the year Lynn Margulis wrote a paper basically saying that once long ago in the primordial soup, a bacteria that could actually use oxygen to make energy got taken up by what eventually became us, human cells. And so now think of a human cell with these former bacteria that know how to make energy inside them. But what's been going on for almost as long as that is viruses have been attacking bacteria. And what happens is the virus inserts its genetic material into the bacteria, and then the bacteria fights back with its own immune system. If you can believe it or not, the bacteria have an immune system. And in fact, that system, which is called the CRISPR-Cas system, CRISPR-Cas chops up the viral material and protects the bacteria. Human scientists recently used that discovery of the CRISPR-Cas system and made a Nobel Prize-winning discovery about how to edit genes and cut genetic material to fit your needs. But that's a pathway long ago in bacteria. So we're now set up that when this virus comes in, and SARS-CoV did the same thing, we can look with big genetic screening programs and just say, when you get an infection, what happens to your cells 2 hours, 12 hours, 24 hours after infection? And what you're seeing is this virus is basically turning thousands of our genes on and off, and it's making proteins which are toxic to the cell.
Gerald, I want to come to you in terms of how we can use this research not only for other viruses, but with long COVID. That is a big player here when people are infected and they're suffering not for months, if not years. Can this be applied?
Absolutely. And I think this is one of the critical things. So as many people know, we're trying to understand what is it that drives long COVID. As an infectious disease doctor, I've known for many, many years that various infections leave people with prolonged, persistent symptoms that can go on for months and months, if not in some cases years. And so trying to understand what is the mechanism by which that infection causes that problem is really important. So there's lots of speculation. Is it persistent virus? Is it the immune response, which is sort of continuing on long after you've recovered from the illness? What I thought was very critical, and this is speculative, is that if the viruses are causing significant damage to mitochondria, even if that's a reversible process, that mitochondria is the power supply of the cell, as Dr. Archer will tell you very many times. That's critical to how cells maintain their health. And healthy cells are what drive all of these systems in our body that allow us to, to digest food, to think with our brain, to have the heart pump and stuff. So if this mitochondriopathy, this disease of mitochondria, which is caused by the virus, is significant and lasts for a considerable amount of time because of, say, viral persistence, then that will very much help us understand what causes long COVID in particular, and potentially at least what causes a lot of these persistent long-term symptoms that have occurred in people who've been infected with other viruses as well. So I think this actually very, very much excites me into understanding that. And again, I can't emphasize how important the work of scientists like my colleague Stephen Archer is. Understanding how viruses cause disease is fundamental to us being able to understand what those processes are and what we can do as physicians to develop treatments and or understand that whole process a lot better. It's incredibly important.
Stephen, you get the last word on this. Now that we have this information, that you guys have put several months of work into this, how can we better prepare for the future?
Well, for our modest discovery, I think the thing is we show the virus has broken the mitochondria into pieces. We're developing drugs to prevent the fragmentation of mitochondria. We've shown it turns on apoptosis. We're developing drugs, small molecules that attack that. But the one thing I would be remiss if I didn't comment on is the incredible courage of my colleagues, the coworkers who are at the bench. And these 24 scientists, right now, we now have vaccines. Everyone feels a little bit more comfortable. But in 2020, people were afraid. We didn't know, could MHV1 infect humans? We didn't know how safe it was to work with H-CoV OC43. And these people came in when most people were forced to stay home. They were at the bench every day working on this. And they came from India and Iran and England and all over the world, Brazil. And they were so passionate. They are so passionate about their discovery. And I think a couple of things that I'd like to just leave Canadians with listening to this is we need to better fund research in Canada because these people live tenuous lives. They live from grant to grant. If I don't get research funding, they don't have a job. And we need more continuity in science and the ability to go deep and try to invent something. That's certainly something we invented insulin here in Canada, or discovered it, at the University of Toronto. We can certainly do that for new therapies for COVID-19. So that's the main thing I would leave your listeners with, Jeyan, is just I think we've discovered that mitochondria are targets of virus, and we can develop drugs to stop that. Made in Canada science is important, and we bring the best people from around the world because they're passionate about having a chance to do science and work with docs like Gerald, and so I think we should do more of that.
Stephen, Gerald, thank you so much for the work that you do, and thank you so much for joining us on the program tonight.
Thank you for the time. Nice talking to you.
The Agenda with Steve Paikin is made possible through generous philanthropic contributions from viewers like you. Thank you for supporting TVO's journalism.